So, let's begin. We are talking about cells and how they specialize, but everything needs a story. If I just give you the raw, information when there's no context, it doesn't seem important, then the meaning is not as meaningful. Okay. So, let's give it some context. Some of you may have heard of the disease mono, which is short for mononucleosis. Mono affects different people in different ways. I like that statement. And it is a disease that can make you sick. And for some people, they'll get bedridden. For some people, it's kind of like COVID. Some people, they kind of, they get, diarrhea and a little bit of nausea and they're a little sick. And some people, it rips through their lungs and they go into respiratory distress. They're in extreme pain and they might even die from it. Mono's not normally, and it's not a deadly disease. But there's different effects depending on who you are. So when you look at a normal person's throat, if you're feeling the nausea and you are really sick and you can't swallow your food and other things like that, then you'll go to the doctor, they'll do swabs, but often they'll do certain things, including looking at your throat. So in a normal throat, if you say, you'll see the uvula dangling at the back and you can see lots of pink back there. But if you have mono, then you'll see that there's this whitish, grayish stuff at the back of your throat. And what's happening is you're seeing your tonsils typically enlarged, they're inflamed. And so you want to treat this. Now, the problem with mono is that its effects are very similar, or symptoms are very similar to other things. If you look at the symptoms of headache, sore throat, fever, enlarged neck lymph nodes, that sounds very similar to other things. It might even sound similar to COVID and flus and things like that. So how do we determine that you have mono? Well, they can do a bacterial test, but imagine this, using a biomarker. A biomarker is a small measurable substance. It's a molecule or an enzyme or DNA or RNA or cancer or protein. It could be any one of these things. And what these biomarkers do, they are typically going to be indicative of some phenomena such as a disease infection, like mono, or environmental exposure to radiation, let's say. A biomarker is a biological characteristic that is objectively measured and evaluated as an indicator of normal or pathological, which is disease-ish, processes. So, if you have mono, then there's very good chance that your cells are releasing a chemical that you could detect, a biomarker that could be measured to determine that you have mono. So that's pretty cool. This is the same technology that they want to use to be able to detect things like cancers before it becomes a problem. Now, let's say you do have some kind of genetic disorder, as opposed to just a disease, but genetic disorder, and a biomarker is able to tell you this. Well, for you to fix this problem, genetically, you generally have to fix every single cell. It's very hard to focus on one part of the body, so you're fixing everything. And if you make one chromosomal mistake, we talked about this, Down syndrome, that's a big issue. In grade 12 biology, if you make one little mistake with the nucleotide, you can end up with cystic fibrosis or sickle cell anemia. These are not good, right? So cystic fibrosis, what this is a genetic disorder. You're born with it. And in a normal person's airway, they have these nice clean straws that allow air to flow up and down your respiratory tract. But with a person with cystic fibrosis, they have some kind of inflammation infection in their alveoli and the little tiny bronchial tubes that feed into the alveoli, and they get filled with liquids, right? This liquid is a mucus, and it makes it very difficult to breathe. So imagine you're breathing through a giant straw. Easy. Now let's take that giant straw, make it a normal straw, and you just run 100 meters, and you're trying to breathe through that little tiny straw. It's very difficult. It's kind of like an asthma example I'm giving you here. So cystic fibrosis, it's all down here. So it's very difficult for them to get air in the lungs. It's very difficult for the respiratory process to proceed. So how do you fix this problem? Because this is a genetic issue. There are different types of cystic fibrosis. Some cystic fibroses are treated with different types of drugs. And it so happens that ivacaftor is a drug that specifically targets one type of cystic fibrosis mutation. It only will work on 5% of the mutations. That's it. The other 95% are a different type of mutation. So this graphic that you're looking at here represent several different types of cystic fibrosis mutations. And so this drug works on 5%. So it's great. That 5%, they take this drug, cystic fibrosis problems, done. No issues whatsoever. That's great. Okay, so what happens? What can you do? We've talked about, well, we haven't talked about CRISPR yet, and we'll talk a lot about this in grade 11 and grade 12, and based on our timeline, I can't talk too much about it. But this is a genetic technology that allows us to go into the genes, go into the cell, and modify the genes. We can edit the genes. And so if there's a mutation, we can edit the mutation out. We can fix the mutation. Right. If you're trying to fix 1 trillion cells, though, you want to make sure, once again, that you're not making a lot of mistakes. So now we get into the realm of precision medicine. Consider this drug called explodinol, right? Explodinol. And it turns out that when you use explodinol, that 5/6, so for every 6 people, 5 people are completely cured of the disease. Perfectly fine. Explodinol cures cystic fibrosis. Done. So you're like, yes, let's put it on the market. But let's say 1/5 of those people who take the drug, they blow up. That can happen. So even though 5/6, 50 out of 60 people could be completely normal with this drug, because 10 out of 60 might blow up, we can't use it, despite the benefits. It's not going to work. So now imagine a microRNA biomarker, a biomarker that differentiates between people who are explodey patients and people who are non-explodey patients. And so there's a marker inside your body that you can measure, and you say, you know what, you got this marker, there's no way you explode. We'll give you the drug. And now you are cured. You have the marker, sorry, we cannot give you this drug. It's going to be problematic. And this is where precision medicine comes into play. It allows to look at very specific people and precisely diagnose and then give an appropriate prognosis that gives them a certain type of medication that will make them better. And that medication that works for them might not necessarily work for another person. Okay, precision medicine. So this is the world that we're entering. We're entering the world of precision medicine. More cell technology includes genetic engineering. You've probably heard of this. And this is where we modify the genes of one species so that it can be inserted into another species. Right? There's a lot of different forms of genetic engineering. This is the one we're going to talk about here in grade 10. And so in order to modify a gene, we typically will recombine it with something else. Okay? Now, at this point, if you haven't already, you should have opened in the document. You will notice that it asks you to open Nearpod, right, in the Google Doc that you're following along. And when you open up Nearpod, you're going to see a reference specifically to today's lesson, this week's lesson. And so that Nearpod, as you work through it, just follow along. And you're going to see some slides that are very similar. As you get later in the presentation, you'll see exact slides, and then you're going to see questions. So hopefully you're working through this as well with me. All right, so genetic engineering. So what genetic engineering allows us to do, it allows us to modify organisms. And we can then make these organisms better for us. We typically don't modify organisms for them, we modify it for our benefit. So I want you to know at least one transgenic example. On the screen here, I have a couple of examples. Herman the Bull was a cow that was modified, and people were not happy because here we had a genetically modified cow and that wasn't natural. There's a lot of things online that talk about genetically modified organisms and the good or the bad about it. But the truth is, if you're shopping in 2020 in a store, pretty much anything you buy, or if you go to the farm, anything you grow, typically nowadays, it's not the same as it was 50, 70, 100 years ago. We've modified most of our food. The corn is actually a modified grass. The watermelon is huge. Those strawberries you get, that's not natural. And most of our animals have all been genetically modified in some way. All right? We also, scientists, will genetically modify mice and rats to remove their immune system so they can explore what happens when you expose them to certain diseases, or what happens when you try to graft in human parts. So this mouse here, I encourage you, if you look, it's interesting to you, to Google it, this mouse with an ear on its back. What's happening here? A specific example of transgenics that worked effectively. This has been ongoing. This is something that they figured out, a decade ago. But it's a lot of people, they're not sure about this genetically modified story, and so they won't accept it. But in certain parts of the world, vitamin A deficiency is huge, and if you don't have vitamin A when you're growing, especially, then you end up with certain developmental disorders. And this is particularly a problem in Africa and Southeast Asia. Golden rice has been transgenically modified. So they took this gene and modified it so that this rice now produces vitamin A. And if you take it, you'll not have any vitamin A deficiencies. So this, just recently, I believe in the past year or the past year and a half, the government in Southeast Asia, they finally said, you know what, this is a problem and this could be a solution. Okay, let's give this a try. Right? So golden rice. Another example here is cheese and rennet. Yeast fungi or bacteria modified to produce the enzyme chymicin, which splits milk to make cheese. Traditionally, rennet, which is found in your cow's stomach, is used to clot cheese. However, the demand for firm cheeses surpassed the amount of rennet available, and therefore they needed another source. And so they were able to recombine chymicin so that it is able to produce it without the cow. Right? So a lot of cheeses are produced genetically, they're modified. In many cases, we are trying to figure out how to beat cancer. And so a lot of our drugs that we use to experiment and hopefully fix cancer are grown in other organisms. And for us to grow them in other organisms, we need those other organisms to express human-like factors. And so those human factors are spliced, basically, are recombined into other organisms. The vaccine against non-Hodgkin's lymphoma is grown using mice and the tobacco plant. I remember my youngest, Darian, when he used to get his needle when he was like 2, 3, 4, 5, he was loud, he screamed. I was working at school, Christine, my wife, would bring him to the doctor, and everyone knew when Darian was getting a needle because he would scream. So if we were able to put the vaccine into a fruit and make the vaccines edible, imagine, right? So we could genetically modify a banana or tomato so that it had the ability to give you the hepatitis B vaccine or a cholera vaccine. That would be great. It would be easily accessible, more people could get it, and less people would get sick. Gram for gram, spider silk is very strong. If you compare it to a steel rod and you get spider silk and wind it up into the width of a steel rod, the silk, the spider protein, would be stronger. So imagine if you're able to build something with this. But anyway, spiders only produce so much. So one of the things that they're able to do is they've got these goats, and goats produce a lot of milk in their, and when they produce their milk, there's a lot of protein in their milk. So there was an FDA approved human antithrombin protein, and it's used to treat a rare blood clotting disorder in humans. So goats have also been genetically modified to produce spider silk. So they've inserted a gene that allows the goat to produce spider silk. They milk it from the goat, they go through a process, and they've made bulletproof vests from this protein. I mean, how cool is that? And there is a huge shortage of transplants. There's people out there waiting for kidneys, livers, hearts, you name it, all sorts of different body parts. And if we were in school, in May, we were supposed to have a visitor who had a transplant, along with a nurse from Toronto General Hospital that works at the transplant hospital, and they were going to explain the process. So we don't have that option anymore. There is a big shortage. There's people who are dying because they're not able to get these transplants. There's not enough people. So what would happen if we could genetically modify a pig with human organs? This is called xenotransplantation. And this was a process that had been explored, but then because the pig has this particular virus known as PERV, it's a porcine retrovirus, when it comes into human, it makes a human sick. So they weren't really able to beat this virus. So they kind of gave up on it. But lately with the CRISPR, the use of CRISPR as a genetically modifying tool, they have a lot of hope that xenotransplantation might be more possible. So the idea of growing pigs with human organs, taking the organs out of the pigs and putting them in a human, that could greatly reduce the waiting time that people are on the list for in order to get a life-saving transplant. Okay, so when you go through the Nearpod, one of the questions that's going to come up, it's going to ask you, when you hear the word stem cell, what comes to mind? So what comes to mind? So go ahead and complete that. You should pause this now. I'm going to keep on talking, and I'm going to explain what a stem cell is. Now, here's a series of questions. Again, as you work through the Nearpod, so you're working through the Nearpod at the same time I'm going through this lesson, and you're going to see this slide, and the very next slide, you're going to answer the question. Which one is true? Who is correct? Tissues are the basic unit of structure, and organs are the basic unit of function. Cells are the basic unit of structure, and organs are the basic unit of function. Cells are the basic unit of structure and function, or is it Ray? Organs are the basic unit of structure and function. One of those are accurate. So I'll give you a moment. Don't move with me. Pause it, answer the question, and then move. All right. So cells are the basic unit of structure and function, as you probably should have predicted based on the story of cells that we've done for the last 3 weeks. So 5 things that you've used today: spoon, chair, toothpaste, fork, bowl. What have you used? So again, pause while you think about it. Hopefully maybe you're on with a partner or someone else who's kind of on the screen with you. You guys can talk about it at the same time. That'd be a really effective way of running through this. How does the structure of these things help? So if I were to grab a spoon, a giant spoon here, how does the structure of this help with this function? If I'm going to use this to eat my cereal, which has liquid milk in it, then it's got a nice bowl to hold the milk. It's got a nice surface here that I can have my Fruit Loops in it without it spilling out, and then I can put it in. It's got a handle that's ergonomic so I can grip it well. So the structure supports the function. All right. So you should know that cells have different roles in your body, and their structure helps with their function. Okay. So when you take a look at these cells, what are they? So you're working through the Nearpod. You're going to answer on the next slide what you think they are, and then I will tell you that they are red blood cells. And red blood cells carry oxygen. It's very important that they carry oxygen from point A to point B. As a donut shape, it's the perfect shape. It allows them to move through a tube without getting stuck. If it was anything with a corner, they might jam up on each other and clog up the blood vessel and prevent oxygen from being delivered. What about these cells? What are they? Go ahead and go to the next slide where it says, what are these? Answer it. Pause if you need to. So these are neurons. They are communications. They send information from the brain, let's say, down the spinal cord to my toes. So they have a perfect structure for the function of communicating information from point A to point B. I'm sure you recognize these ones. Go ahead and type it in if you need to. So these are sperm cells. And sperm cells, their job is to deliver the DNA from the father to the egg. That's it. So they need movement, they need a tail that's going to push them in a certain direction, and then whatever makes it there first does the job. This one here is particularly interesting. So let me show you a picture of this in action. I should have had this activated so you guys could see it. This is it's not the true color, but let's look at a picture of one of these. Okay, so here it is. Let's rewind back to the beginning. So maybe you can guess what this is. This is a white blood cell inside your body chasing a pathogen, some bacteria, and at some point it's going to capture it, surround it, and destroy it. So this is a part of your immune system. This is the same thing that's going to help you fight COVID if you happen to contract it at some point, or a flu, or chickenpox, or any of so many other diseases that are a possibility.